for the invitation. It's a great pleasure actually to be able to share some of the data that we're going to be actually published online soon on this new NAD precursor, Trigonelline, as you mentioned. And this in particular is work that I want to highlight, also the contribution that came from my previous appointment when I was working in Switzerland with Nestlé Research. So actually this work is a beautiful example of also how companies and academic environments can work together in order to bring translational findings closer to the both scientific community, but also to the public. So without further, let's say, delays, I want to introduce you some of the key findings that came up from our research on Trigonelline for muscle benefits and longevity across species. So just to give you a bit of a context about what is sarcopenia. So sarcopenia is basically a loss of functional let's say, outcomes in muscle, not just in terms of muscle mass, but also really in terms of muscle functionality, so ability to move, strength, and so on. It's, of course, an age-associated disease, so its prevalence is increasing with the fact that the most advanced civilizations and groups in the world are, of course, becoming aging or super aging. And this is, for instance, very true for Singapore, for instance, where I'm currently working. But from a molecular perspective, people that are familiar with the aging field, they may be aware that the aging process from a biological standpoint is composed of several intertwined hallmarks, which are essentially declines in the different cellular functions. And from this point of view, muscle aging and therefore also muscle sarcopenia are kind of sharing some of these hallmarks of aging and disease. For instance, we know that for muscle aging and sarcopenia, there is increased inflammation in our muscles, dysfunctional mitochondria, which are the centers for producing energy and important metabolites in our cells, increased oxidative stress, also increased fibrotic damage within the muscle, compromising essentially the muscle elasticity and ability to function. And very importantly, there is also dysfunction of satellite cells, which are the muscle stem cells, which are important for supporting muscle regeneration or even in maintaining muscle mass in that sense, and imbalance in protein turnover, meaning that there is tendentially and often in muscle aging and sarcopenia more muscle catabolism, so loss of protein rather than synthesis. So from a clinical perspective, how do we measure sarcopenia? There are three at least clinical outcomes that need to be kept in mind for the rest of the discussion that we used also in our studies. And these are aligned essentially between different consortia. This is for instance, we have the European consortium, and here is below you can see the parameters measured from the Asian consortium. And actually, these are basically the three same parameters, which are gait speed, grip strength, and appendicular lean mass. Meaning, if your values decline below these threshold values that are indicated here, people will be scored or diagnosed as sarcopenic. Now, another key molecular factor that has been coming up in the literature from different studies, including from my own research, but also from very famous groups worldwide, such as, for instance, David Sinclair or Johan Auwerx and so on, have actually highlighted the key role of a coenzyme that is involved in more than 500 reactions in our bodies and pretty much involved in in most of the cellular hallmarks of aging that I highlighted before, and this is called NAD+. So NAD+ has been shown to be important to support mitochondrial function, to support also redox reactions, reduce inflammation, favor DNA repair, and so on. And what people have really focused on, including us, both at the preclinical level and more recently, people are starting to understand how at the clinical level with exercise or lifestyle or supplementation, how we can actually use certain approaches in order to maintain NAD, and what kind of benefits actually supporting or maintaining NAD levels can bring to the different parts of our bodies. So basically through exercise or diet or even using NAD boosters, supporting NAD has been shown preclinically at least to improve pretty much every organ in your body that you can think of, not just during aging, but also during age-associated diseases. And all this, it's basically summarized in this beautiful review from David Sinclair in this case. Now, keeping in mind the important role of NAD as a key coenzyme for most of the cellular hallmarks of aging, and the fact that during the aging process, muscle mass declines, rises, and also the other hallmarks of aging are starting to be evident. What was done before in Nestlé Research, before actually I joined the company, there was this study published in 2019 where what the study did, they did an observational study where they compared in a Singaporean cohort, actually, in male Singaporeans, either healthy elderly or age-matching individual with sarcopenia, assessed with the three parameters that I mentioned before, so grip strength, gait speed, and appendicular lean mass. And what they did, they took muscle biopsies from these patients, and then they looked at different molecular signatures in these tissues. What emerged is that on the other end, on one end, mitochondrial transcription pathways that were completely downregulated, and also OXPHOS, indicating proteins indicating mitochondrial respiration complexes that were completely downregulated, indicating mitochondrial dysfunction. And very importantly, what was also measured in a few biopsies that were still available from these patients is that NAD+ levels were significantly lower in the muscle tissues. Also, very importantly, if you look at the muscle function, so again, the three clinical features of muscle health, these were actually positively correlated with the muscle NAD, meaning that higher muscle NAD correlates with better muscle health overall. And this kind of resonates with what I just mentioned before, with NAD being very essential to support normal homeostasis and functional outcomes as we age. Now, at the time I joined and I started leading the group on this project, what we also had available at the time were actually serum samples from the same cohort. So what we wanted to do there is to look at through different metabolomics analysis, what kind of metabolites were changing at the systemic level, reflect what we had just observed for sarcopenia. So could we identify either new biomarkers or new metabolites that could be used as targets or therapeutic interventions for muscle aging or sarcopenia? We looked at different metabolites, including different kind of vitamins. So we measured also on a platform all kinds of vitamin B3 related families members, which are essentially the precursors for the biosynthesis of NAD+. Of all these molecules, the only one that was significantly changing, and it was downregulated in sarcopenia actually, was this molecule called trigonelline. It also turns out that if we looked again at the three clinical outcomes of muscle health, so lean mass, grip strength, and walking speed, serum trigonelline levels were positively correlated with muscle health. So as we saw for NAD levels in muscle, serum trigonelline levels were also positively linked with better muscle. And also when we looked now trying to link what would happen if we would look at higher levels of NAD in serum with genes that would be modulated in our muscles, what it turns out is that the most positively enriched pathways in muscle, when we have high levels of trigonelline, it was actually a mitochondrial OXPHOS, so mitochondria-related pathways for energy production in our cells, which makes, in that sense, if we think of possible link with NAD, that would make completely sense, because NAD and molecules that support NAD production, they're also generally linked with better mitochondrial function. So with this in mind, we decided indeed to look at what is trigonelline. So how does it look like, and how it could be related to mitochondrial health or with NAD metabolism. When we look at the structure of trigonelline, it turns out that it's pretty much a methylated form of nicotinic acid, as you actually had anticipated before, Michael. And from the literature, the only things that we could find from the past, and this is very old findings from trigonelline, is that it seems to have beneficial effects from the metabolic perspective. So it seems to work preclinically in, for instance, models of diabetic diabetes. It can lower plasma and liver cholesterol in NTGs, for instance, in preclinical models of NASH, of liver disease, essentially. It can improve glucose transport, but essentially we didn't find much else that would link it specifically to its mechanism of action. When we looked at the origin of this molecule, so where is it present? I mean, how do we find it in nature? It turns out that it's mostly produced by plants, and to some extent by it's found also in the human gut microbiome. But really, in the majority of trigonelline you can find in nature is present in plants, in particular in fenugreek seeds and coffee beans. So things that are part of our basically regular nutrition for people that, for instance, drink coffee. And the only other thing that we could find from literature in the past, it's a very old study from a Japanese group that showed that if you would take liver extracts from different animals and you would add trigonelline to these liver extracts, you would get demethylation of trigonelline into niacin. So with this hypothesis in mind, we wanted to actually start looking whether trigonelline could be comparing like an NAD precursor that could be compared to other NAD precursors. Before we go into the effect on NAD, we also wanted to actually understand how the potential of using this molecule for human application. So one important parameter is actually for all these molecules in general, for supplements, but also for drugs, it's their stability. We know that, for instance, for NAD precursors such as NR and NMN, they're actually, they've been shown to be effective preclinically, and there is clinical trials ongoing. However, these molecules are known to be interconverted into, for instance, degraded into nicotinic acid or into NAM by our gut microbiome, and they are rather unstable in the human serum. As you can see here, we did a time course just spiking the molecules into human serum at 37 degrees, and we follow them over time. You can see that NR and NM, which are part of the salvage pathway of NAD biosynthesis, they actually very rapidly disappear from the serum in that sense, because actually they get degraded and they release actually NAM or nicotinamide, which is also part of the salvage pathway of the NAD biosynthesis. But it's considered to be, to some extent, also a degradation product of NAD. So this it's now quite very established also with our assays, and it was already reported by other groups before. Importantly, for trigonelline, being just a methylated form of nicotinic acid, that's not the case. So trigonelline is very, very stable, even more than 24 hours in serum. And of course, it doesn't release, it doesn't degrade into NAM just in a, let's say, a body-free system, just in a impure serum. So with this in mind, and so knowing its stability and knowing its link now with mitochondria and possibly with NAD in muscle, we wonder whether trigonelline could actually act as an NAD precursor in animal species and impact longevity and muscle health. So we started by doing very simple assays. So the proof of concept is whether if we supplement trigonelline to different model organisms or in vitro systems, whether we can increase NAD levels. That was the case. So when we, for instance, provide trigonelline in different doses to HSMM cells, which are primary human myotube cells, so derived from human donors, we can see that dose-dependently trigonelline could increase NAD+ levels. This is true also when we use primary human myotubes, not only from healthy patients, but also from sarcopenic patients, meaning that the conversion of trigonelline into intermediates that can lead to NAD biosynthesis is still occurring even if you may have sarcopenia. And that's actually important to know from a translational application perspective. We then also wanted to use simple model of aging, and this in vivo can be done, for instance, with nematodes like C. elegans, which age quite rapidly, and you can supplement them with drugs, for instance, and then study mechanism of action of the drugs. In this case, what we did, we compared the NAD levels between young and old worms, and you can see that if we take day 8 worms, which are already aged, they show a decline in NAD, which was significantly rescued by supplementing trigonelline at 1 millimolar for 8 days in these worms. But more importantly, we wanted to then validate the possible effects in vivo. To do so, we took, in this case, still young mice, so 3-month-old mice. We gavage trigonelline 300 milligram per kilogram, and this is a quantity of an amount of trigonelline that is equimolar to NR, for instance. So this means that we are using what people use for NR when they do studies in rodents in order to increase NAD and bring benefits to their mouse system. So with this in mind, we did a gavage for 2 hours, and then we collected different tissues and measured NAD+. What you can see here, if we look, for instance, at liver, muscle, kidney, and blood in this case, trigonelline significantly increased NAD levels after 2 hours in all these tissues. One question that we have actually got quite consistently, and that's why I'm showing now also this new data that are part of the story that will be published soon, is what happens actually, what are the values of trigonelline itself? So how bioavailable is trigonelline when we look now at the same tissues where we have seen increase in NAD? So what we did here, again, we took the mice, we gavaged them with the same amount of trigonelline, and then we measured trigonelline with a mass spectrometry with absolute quantification, either at 2 hours, or we led essentially like a washout period of overnight, meaning like more than 12 hours in this case. What you can see here is that depending on the tissue, you may have already baseline levels of trigonelline, which can be either very low, so we are here in the low micromolar range, or they are even below limit of detection, for instance, for gastrocnemius in muscle in this particular case. But upon supplementation with trigonelline at 2 hours, we get a pretty strong increase, significant increase in trigonelline in all the measured tissues, including in muscle, in kidney, in liver, and in plasma. And of course, a huge excretion of trigonelline in urine. This actually has already been kind of described in other studies, also in humans. But nevertheless, after 2 hours, we are able in all these tissues to reach quite high, actually, micromolar range, especially in plasma. We are in the range of 100 to 250 macromolar, for instance, after 2 hours. But you can see that after a washout period, we almost reach baseline levels again. In most of the cases, in liver, for instance, we still retain a significant increase of trigonelline, but for instance, in muscle and in plasma, we almost go back to baseline in this case, meaning that the washout is quite quick. It still retains trigonelline in most of the tissues, but for, let's say, supplementation purposes, this is useful information that will help us to decide or define in the future whether how frequent or how much trigonelline can stay in our body and how frequently we may want to supplement it or not. But now let's go to the mechanism of trigonelline. So we said it's a methylated form of nicotinic acid, but nobody had actually shown that it could actually work as nicotinic acid. So that was the purpose of these experiments that you see here in cells. So here, what I'm giving you here is a simple cartoon of the NAD biosynthesis pathways in our bodies. You have the salvage pathway, which I mentioned before, which makes use of nicotinamide or NAM. And nicotinamide gets usually converted into NMN, which is another known NAD precursor currently already available as a supplement. And of course, you have nicotinamide riboside, which also can be converted into NMN. And then this basically can then lead to the formation of NAD+, basically with 1 to 2 reactions in this case. And then you have the press-standard pathway, which is the one that nicotinic acid uses in order to generate NAD through several reactions. And what I want to also bring you to your attention is that for all of these pathways, there are several key enzymes that are important for the conversion of the intermediates into the NAD pool. And some of these enzymes, like MPRT, which is the key press-handler enzyme to convert nicotinic acid, can be inhibited by 2-hydroxynicotinic acid. Or NMPT, it has a specific inhibitor called FK66, which can be used to block essentially the salvage pathway. And in this case, NR being downstream of the NMPT and also independent of the press-handler pathway can be used as a sort of positive control in order to increase NAD independently of inhibition of these pathways. So that's what we did here. We went back to our muscle cell line system, and we simply provide similar amounts, 1 millimolar of trigonelline and nicotinamide riboside, and you can see that both very significantly increase NAD levels in these cells. What happens now when we provide 2-hydroxynicotinic acid and we block the press-handler pathway? Trigonelline doesn't work anymore, but NR, which is independent of this pathway, actually can still significantly increase NAD levels. But what happens now when we combine FK66 to block the salvage pathway with a 2-hydroxynicotinic acid to block the press-handler pathway? What you can see here, first of all, is that FK66, so blocking the salvage pathway, induces a very strong NAD depletion phenotype in the cells, but with trigonelline or NR, which are both independent of the salvage pathway, with FK66, can actually both rescue completely with trigonelline and even boost further with NR the NAD levels, even in the presence of the inhibitor. However, when we now combine also with 2-hydroxynicotinic acid and we block the press-handler pathway, you completely lose the effect of trigonelline. Again, proving that trigonelline works mostly through the press-handler pathway. We have done also metabolomics analysis in order to prove that the downstream metabolites of the press-handler pathway are increased following trigonelline administration, and that turns out to be the case. What happens now when we want to look at functional outcomes in cells? So as I mentioned, NAD+ is very important to support mitochondrial function, for instance, through the action of sirtuins and mitochondrial biogenesis. So one thing that we wanted to observe is what happens to mitochondrial function when we deplete NAD and when we rescue NAD with our NAD precursors. What we did here, we measured mitochondrial membrane potential or maximal respiration through Seahorse methodology. You can see that when we use FK66 again to block the salvage pathway, as we saw for NAD that goes down, also mitochondrial membrane potential declines and mitochondrial respiration also declines, which is in line with what we would expect from the interdependency between mitochondria and NAD. However, again, now blocking only the FK66 means that when we use NR or trigonelline, we can actually rescue completely the mitochondrial membrane potential in this case. However, when we now block the price-standard pathway on top of the salvage pathway, trigonelline is not effective anymore in this case, while NR can still function pretty well. And the same happens with mitochondrial respiration. Again, trigonelline can rescue the inhibition of the salvage pathway, but not when also the price-standard pathway is inhibited. Also, this is something very, it's an observation I want to bring to our attention, is that differently from NAD levels, where you have seen that NR could very potently induce NAD levels, even more than trigonelline in this cell line, when we look at functional readouts on mitochondrial health, like respiration, mitochondrial membrane potential, so downstream of the NAD boosting effect, actually the effects between NR and trigonelline are quite comparable. And we have observed actually the same also when we go in vivo with nematodes, for instance, when we look at lifespan or muscle integrity. So what I want to really bring to our attention is that even if NAD boosters may affect NAD levels differently, when we look at functional efficacy of these precursors, in this case mitochondria, but it could be anything, could be mobility, muscle strength, or something else, it's very important to keep in mind that these compounds may actually lead to comparable efficacy. And that is very important, meaning that we need to also know the biology of these compounds in order to understand which cohorts or applications may benefit more from using different precursors. So next, we wanted to move actually to observing benefits of trigonelline in a more functional kind of readout setting. And for this, we went back to an accelerated model of aging, if you want, or a short-lived animal like C. elegans. Why? Because in these animals, basically within 10 to 11 days, like in this case, you can observe a loss of muscle integrity. So these are muscle fibers in young adult worms versus day 11 adult worms. You can see the loss of muscle fiber integrity. But following 10 days of supplementation with trigonelline, we were able to maintain, to a certain degree, more muscle integrity, more integrity of the muscle fibers compared to day 11. This was reflected by an improvement in mobility of the worms during aging. You can see here 2 time points that the paralysis of the worms is actually lower when the worms are treated with trigonelline. And importantly, this reflects also on lifespan in these animals. It's very, these are short-lived animals, so you can see that the control worms, they live maximum approximately 24, 25 days. However, with trigonelline, we are able to extend the lifespan up to about 30 days with a median increase of about 17.4%. But this is not true when we actually inhibit or we repress genetically the expression of MPRT in the worm, which is essentially the key enzyme that is important to use trigonelline or niacin. So this also proves that also in vivo, there is a dependency of trigonelline with the price-standard pathway. So if we cannot use the price-standard pathway, we lose the majority of the effects of trigonelline in this case. And finally, we wanted to actually test benefits now in an in vivo model during aging. And in this case, we used, in collaboration with Gordon Lynch in Melbourne, we made use of aged mice, and we treated them chronically with trigonelline. So what we did, in particular, we took 2-year-old mice, which were fed either a controlled diet or a trigonelline supplemented diet, again, with the same quantity that we used also for the gavage experiment. And we supplemented these mice for about 3 months. And then at the end of the study, we measured things such as grip strength and in situ muscle force, which I will explain in a moment. So at the end of the study, we collected the different tissues, and then we looked at things like, for instance, limb mass, and then we measured the phenotypes for muscle health. So one thing that emerged from this study is that the limb mass actually in these mice at the end of the study was not impacted. It's also true that mice compared to humans do not necessarily become sarcopenic very easily. So you really need geriatric mice, meaning much older than 2 years old, in order to start observing some consistent loss of limb mass. And also an important thing that we did here, we measured the peak force in the tibialis of the anterioris of the mice, meaning that we put the mice to sleep and then we basically use electric stimulation in order to cause the maximal tetanic contraction in the muscle of the mice. And what we observed is that the peak force in these mice was not changed at baseline, meaning that the maximal force, even if you use trigonelline in these mice, is not going to increase. However, what we did here, using the same setup, we now started to stimulate the muscle with several repeated contractions over time. So in order to induce muscle fatigue. So in humans, that would be a little bit like if you were to lift weights, for instance, and do repetitions up to a point where your muscle get fatigued, then you don't feel that you can lift anything anymore, right? So that essentially your muscles are not able to contract well anymore. So what you can see here is when we do this in rodents, you have here in gray, this line is for a young cohort, so it's our reference cohort. And then we have an aged cohort here, you can see in black, where you can see that the percentage of initial force is declining following the repeated contraction. You can see a more significant and steeper decline compared to young mice, which is expected during the aging process. However, the mice that were treated chronically with trigonelline were able to maintain an intermediate level. So we were actually able to improve the amount of force that we were able to generate in spite of the muscle fatigue protocol in this case. And importantly, this was also mirrored by a small but significant increase in grip strength in the mice before essentially sacrifice, or in living mice in this case. So with this, what we can say is that trigonelline, in spite of not modifying the peak force at baseline, and there's certainly more experimentation to be done, is that trigonelline levels decline in a human sarcopenic cohort and associated with mitochondrial function. Trigonelline is an NAD precursor across species through the press-under pathway, and it seems to be well tolerated, at least in rodent studies, and to promote muscle resilience to sustain contractions in aging rodents. Another piece of data I just want to show you before we conclude is that our cohort in Singapore was based on a very small cohort of individuals, but we have been able to replicate in a larger cohort. This is an Iranian cohort with more than 100 individuals. We have been able to replicate the association between grip strength and serum trigonelline levels, which we had seen in the Singaporean cohort for trigonelline. And also importantly, we don't see in this case, we have actually also diet information from these individuals. What we don't see is an association between caffeine intake and vitamin B3 intake in this case, meaning that the effects that we see, the association we see between trigonelline and grip strength is not. Although we have seen a partial association with things like folate and fibers, meaning that probably there may be a connection between trigonelline and the one carbon metabolism or the folate cycle, for instance. However, we don't have now at the end data that this would be something that needs to be investigated further. And along with the other questions that I'm highlighting here about the demethylation process of trigonelline, which for mammals is not established, and at this point, whether supplementing trigonelline would have a real impact for muscle health in humans. So I think with this, yeah, I'm done with my presentation, and